for eukaryotic cell wall, so most eukaryotic cells have cell wall that are much simpler than prokaryotic. In fact, it is the cell wall that allows the scientists, the researchers, to quantify some of the eukaryotes. So for fungi, they have ergosterol also in their cell wall, and this is the component that mycologists use to quantify the biomass of fungi in environment. Okay. Chitin is also quite specific for fungi, but the component that usually researchers use to quantify the amount of fungi in the environment is ergosterol. Then, other than the cell wall, there's glycocalyx. Glycocalyx is just the sticky carbohydrates that surrounds the cells. So, for cytoplasmic membrane, this is to carry out movements across eukaryotic cell membranes. For example, you've already learned simple diffusion, facilitated diffusion, osmosis, and active transport. So these are the methods that the eukaryotic cell transfer components from outside into the cell. So group translocation does not occur in eukaryotic cells but occur in prokaryotic cells. Okay. So you've come across these terms, phagocytosis and pinocytosis. So the main difference is if there is a particle during the engulfment process, that is phagocytosis, which means the eukaryotic cell is eating up another particle. But if it's just solution, then it is called pinocytosis, or cell drinking. Okay. Then you also have receptor-mediated endocytosis. So these are for special components. So in the cytoplasm, you have skeletal tissues, okay, consisting of microfilaments, then intermediate filaments. So you can look at the diameter differences. So this is thin, this is thicker, and this is the thickest microtubule. So all this play a role in providing support, shape, and movement. Okay, so this is the function of the microfilaments, filaments, and microtubules. So usually during phagocytosis, once they detect, like for example, an amoeba detects a bacteria, the engulfment process begins, and then it engulfs the bacteria in a phagosome. But the bacteria is not dead yet until the lysosome lies with the fuse with the phagosome. Then it becomes a phagolysosome. So whatever that is nutritious is absorbed, and whatever that is not is disposed of. Okay, so this is how phagocytosis works. So for nucleus, a specific organelle in eukaryotic cells, it has a nucleolus, which is a dense region within the nucleus itself. It has a nuclear envelope that encapsulates the nucleus, and then you have your ribosomes on the nuclear envelope. So when you do cytogenetics, you will come across how the DNA is actually packaged, okay, into denser and denser form until you see the condensed chromosome. So that's the beauty of design, where a simple double helix can be packaged into a final chromosome. Okay? So the function of the nucleus is, of course, to house and protect cells' genetic information, and also for ribosome synthesis. Okay. Next is endoplasmic reticulum. So there are two types of endoplasmic reticulum. One is rough and one is smooth. What is important is you are able to recognize it. Basically, if you see ribosomes on the ER, then you know it's rough endoplasmic reticulum. And if it's smooth, then it's clearly smoother. Okay. Then next is to know this function. So the function for endoplasmic reticulum, for the rough endoplasmic reticulum, is to synthesize and modify proteins, then to synthesize cell and organelle membranes, and to package and transport proteins that are secreted from the cells. Okay. So the main difference is, for the smooth endoplasmic reticulum, it is more on lipid synthesis and to break down harmful substances. So the main function differences is, for rough, it is mainly for proteins. For smooth, it is mainly for phospholipids and fatty acids. Then for the ribosomes, they are the site of protein synthesis because they carry out translation, and it is present in all eukaryotic and prokaryotic cells. So these two, you must know the difference, okay? For prokaryote is 70S, eukaryote is 80S, and this 80S is made up of 60-40. For 70S is 50-30. Then this is the 16S that we use as a standard for bacterial identification. Okay, so the main function is, of course, to synthesize proteins. It is also available in mitochondria, and this is to synthesize mitochondrial proteins. So when you look at Golgi complex next, so Golgi complex is usually shaped like this, plates. Okay, it looks like the smooth endoplasmic reticulum, except that it is isolated. It is not linked to the nucleus, and it is also in the plates. Okay, plate arrangement. So here you can see that whatever protein that is synthesized will be packaged in the rough endoplasmic reticulum, and when they are ready, you will be encapsulated in the transition vesicle and fused with the Golgi complex. So the Golgi complex role is to receive the protein, then to modify it into its final shape, and before shipping it out. So it can be either as a secretory vesicle, where once it fused with the cell membrane, it will release it out. Okay. So you can see the progress is from the nucleus, the ribosomal translation occurs, then the protein synthesis here, then budding, fused with Golgi complex, and finally exocytosis to release either to use within the cell itself or to exudate it out from the cell. So the budding forms lysosomes, okay? So these are formed from Golgi complexes. So whether it is a lysosome or not depends on what's inside the vesicle. Okay, so for lysosome, it has the digestive enzymes. Okay, so once it has the digestive enzymes, you can term it as lysosome, and of course, The function is to break down macromolecules, to destroy potential predators, pathogens, and also to digest food. This is the function of lysosome. Okay. So other than lysosome, you can also have vacuoles. So vacuoles, you have a central vacuole. This is usually in plant cells. Okay, central vacuole in plant cells. So usually if you see mature plant cells, most of it is empty in the middle, like it looks like empty. So these are the central vacuole. Okay, may occupy up to 90% of plant cell volume. Then you also have contractile vacuole. This is usually in protists or protozoa, aquatic. So when you see the term aquatic, then it has something to do with the floating mechanism, whether the protozoa can sink or float. They can control the density of the cells by controlling the vacuole. Then another one is the food or digestion vacuole, where the function of the vacuole is to, once it engulfs, just like the amoeba you see just now, once it engulfs the bacteria, it fuses with the lysosome to digest the food. Okay, so this shows the different components that we've discussed. The nucleus, okay, where the DNA is transcribed into proteins. Then here it goes to the rough endoplasmic reticulum first, okay, and whatever that needs to be packaged can be sent to the Golgi apparatus or Golgi complex, and the role can be in the form of to help in phagocytosis, or just to as acid hydrolase of food material. So this is to dispose of waste, and this is for drinking, and this is for phagocytosis. So this diagram shows the summary of the different components. Next, mitochondria. So you know that mitochondria is the powerhouse of the cell. This is where cellular respiration takes place. This is where ATP is generated in eukaryotic cells, right? Prokaryotic doesn't have mitochondria. So the site of energy generation in prokaryotic cells is actually the cell membrane itself. So it has the inner membrane, the outer membrane, so you have the intermembrane space, and also the invagination here are called the cristae, and the matrix is the inner liquid. Okay. So it has its own DNA and also 70S ribosome. So this is why there is a theory on evolutionary. There is a evolutionary theory that suggests that mitochondria could be previously a prokaryote that is engulfed by another cell and then slowly loses its own function to become just a powerhouse because of the 70S ribosome that it has. Because 70S is the ones that we see in the prokaryotic cells. That is why there's a hypothesis on evolution from this observation. So the mitochondria in an eukaryotic cell can grow and divide by itself. Okay, so before a cell divides, the organelles will replicate first, so that you have enough mitochondria in both the both new cells. Chloroplast is in plants, is to capture sunlight, essential to carry out photosynthesis, because sun is the only source of external energy for our Earth. and the only the only process that can capture sunlight and then convert it to organic substances is photosynthesis. So it's a disc shape with three membrane systems. You have your outer membrane, you have your inner membrane, and you have your thylakoids. So thylakoids are the plates, the disc. Okay, so thylakoid membranes form the disc. Then the granule is the stack of the thylakoids. Okay, so when you stack up the thylakoids, those are called granule. Singular is granule, plural is grana. Okay, and they are also, like mitochondria, capable of multiplying on their own. Similar reason, before the cells divide, they have to make sure the chloroplast replicates, so that the two new cells will each have chloroplast. Okay. Bacterial growth is unique in a way that they reproduce by binary fission. So this is unique for bacteria, where one is elongated. Once it's long enough, there is septum formation, and then it divides into two. So even before it splits, it will make sure that the DNA, the chromosome, is replicated first. Then each one copy will go to the different ends of the cell before the septum comes in. But this binary fission model that we see is for most of the bacteria, like this. So 1 becomes 2, 2 becomes 4, 4 becomes 8. Okay? However, you must know that there are some bacteria that do not carry out binary fission. You have budding. You also have budding from the hyphae, and you also have stopped organism. For example, the Caulobacter. Okay. And you also have polar growth without differentiation of cell size. What that means is there's no elongation. They split first before they grow. Okay, generation time. This is how you calculate them. This is number of cells. This represents the binary fission growth. So you have exponential growth, and every stage of growth is considered number of generation. So most bacterial generation time is about 1 to 3 hours. So this is dependent upon whether the nutrients are available or not. So this is the formula to calculate the generation time. So by solving for n, by logging it, you can come up to this final equation. So n tells you the number of generations. But when we count, we usually take the cell number from the log phase. So this is the log phase. So whenever once you inoculate bacteria into a new media, a broth, okay, there is a lag phase, initial lag phase. The length of the lag phase depends on how similar the environment is to the ones that is growing before. Because the bacteria has to spend time to synthesize new enzymes, new processes, so that they can take up the nutrients in the new bottle, new test tube here. So the length of the lag phase is dependent on the size of inoculum, the time necessary to recover from physical damage or shock in the transfer, the time required for synthesis of essential coenzymes, and to synthesize new inducible enzymes. Okay, so that's why when you calculate the generation time, usually we take the log 10, the NT time, the number of cells at time t, and time 0, usually we do it here. So we calculate the generation time during the log phase. Okay? We just want to know during the log phase how fast can they double. The mean growth rate constant is this, whereas the generation time or doubling time is the inverse of the growth rate. So once you've reached the end of exponential growth phase, you enter stationary phase. Why is there a stationary phase? Although stationary phase is at a time when there's exhaustion of essential nutrients and it's not optimum for the bacteria, it is actually a phase where the secondary metabolites are generally produced. So most of the antibiotics are all produced during the stationary phase. So if that is your main aim, then you will need to optimize for your stationary phase to make sure that you can prolong the stationary phase so that your amount of product produced is maximum. Okay? So depending on what you are doing, you can regulate, you can control the variables so that you get the most optimum product. Stationary phase is also where spore-forming bacteria will start forming spores. Okay, the death phase is the last one. So death phase. Basically, when there's no longer enough nutrients, the conditions are not favorable, the cells start dying. That's all. Okay, the cell lysis can be by autolytic enzymes or because of the toxic metabolites that the cells themselves produce. The definition of CFU is colony forming unit. That means the colony that forms is from a single bacteria. That means you must ensure good distribution so that the bacteria are located far from each other. That's why we do the spreading, we do the pore plating, and even when we do membrane filtration, we try to make sure they are distributed, well distributed. Because the principle of the method is the colony is formed from a single bacteria. So if you don't spread it properly, you do not fulfill this criteria. So we have done this. The most important thing is to know your range, 30 to 300. Second is to ensure the count ratio is less than 2. So if it's more than 2, take the one at the lowest dilution. Then the rest is just very straightforward. Okay, same goes for filtration. So you can see that for membrane filtration, the sample volume can be very small. Do serial dilution first, and then you inoculate. Okay? Try to keep it to about 1 ml. Even 1 ml is considered small, so usually we will add sterile diluent first before we add the sample. For membrane filtration, whenever the sample size is small, we will add diluent first. Okay? Because we do not want the bacteria to be all stuck at one place. So we add the diluent first, then we add the sample to ensure better distribution. Okay? So that's the thing that you have to remember. So this table is from APHA, American Public Health Association. So they have a standard manual on water and wastewater examination. Hemocytometer is this one. So it's direct microscopic count. So it's a glass slide with a drain. So this is drain for excess sample. So what we do is we just put the sample here, then we place the cover slip. Then whatever there is excess will be drained off. So there is a depth here. Usually it's about 0.1 millimeter. Then from the size of the grid, so usually it's about 1 by 1, so 1 millimeter times 1 millimeter. That's the area. So the total volume that you see is about 0.1 millimeter cube. So this depends on the type of hemocytometer you use. But every hemocytometer will have its specifications. So you must make sure you know. You read first before you use it, so that you know what is the volume here. Which means if you know the volume, and when you look under microscope, if you count the bacteria within the grid, you'll know the number of cells per volume of sample. So hemocytometer is direct counting or culture independent. So it's a culture independent approach to enumerating your bacteria. Because the methods that you've been using are all culture dependent. Right? You have to grow your bacteria before you can count them, enumerate them. So those are culture dependent. So the good thing about culture dependent is you are measuring viable bacteria that's alive. Whereas for direct counting or culture independent, you don't know whether they are alive or they're dead, but you just count them. So there's a difference. Okay? Another disadvantage here is it's very hard to see. Bacteria is small and it's hard to count. Okay? That's why usually we will put some stains inside, methylene blue, just to color them so that it's easier to differentiate. Methylene blue also kills the bacteria so that it doesn't move around. Because if it moves around, it's very hard to count. So next, you also have your epifluorescence microscopy. It is culture independent, direct count. It's of course a very big improvement from hemocytometer because it's kind of like membrane filtration technique where you filter the sample through a filter, but the filter is black in color. It's black in color to provide contrast because the bacteria will be stained with fluorochrome. When it's stained with fluorochrome, it will light up under the microscope, epifluorescence microscope. So it will light up. When it will light up with a black background, it will be very clear and it's easy to count. Okay? So it's a very big improvement over hemocytometer. So the microscope works because it uses a high-powered mercury lamp that will hit onto your sample. It will cause the fluorescence, the fluorochrome to fluoresce. Then when it fluoresces, you can view it with your eyepiece. Okay, so the fluorochrome that you can use can be acridine orange, you can have DAPI, 4'6" 2-phenylindole. You can have FITC, you can have a lot of different kinds of fluorochromes. So there are many different kinds of fluorochromes. The fluorochromes depend on, we talk about wavelengths, because different color, different fluorochromes will emit different wavelengths. So you have blue color, orange, red. Whether you can see the color depends on what filter you have in the microscope. So before you do your work, please learn more about what kind of a microscope you have. Different epifluorescence microscope can have different filter tubes. So don't buy fluorochromes that you cannot use. Okay? Learn what kind of filter the microscope has, then you will know which fluorochrome is suitable. Then you choose that fluorochrome. If not, you will find out that your fluorochrome produces blue light, whereas your microscope can only detect green light. Then it's a waste, it cannot count. Some people also use epifluorescence microscope to determine whether the bacteria is alive or dead by using propylium iodide. So essentially, propylium iodide only stains dead cells, not live cells. So what we usually do is, in research, we stain the same sample on 2 different filters, one with propylium iodide and one with DAPI. So DAPI will tell you total, everything. Propylium iodide will tell you only the dead cells. So this minus this will tell you how many cells are alive. So MPN we've done. So what you need to know is that it's suitable for turbid samples, particles, particulates, samples that are hard to, that are not possible with other approaches. For example, pore plating, spread plating, and the rest. Okay. So it doesn't provide a direct measure. Okay, this is important because it's based on a distribution table, probability table. So it is not a direct measure. That's why the answer itself is MPN, most probable number. So this is for E. coli. This is how you can carry out presumptive confirmed identification of coliform and E. coli. For the enumeration of coliform, fecal coliform in water samples, it depends on what kind of standard approach your lab is using. Okay? There are a few methods, not just one method. Okay? You can do this on just normal McConkey broth at 37 degrees Celsius. If it's lactose positive and gas positive, it becomes your coliform. Whatever tube that's positive, you further inoculate a fresh McConkey broth. But this time you incubate at 44.5 degrees Celsius, and that will tell you whether they are fecal coliform or thermotolerant coliform. And from the tubes that are positive, you can do a streaking on eosinethylene blue. It should give green metallic sheen. Then it will be presumptive. It will be confirmed from E. coli. But if you do not have so many incubators in the lab, then you can do the laurel tryptose broth for the first stage, and then the brilliant green lactose broth for the second stage. Then finally, also EMP. So both these approach still use lactose as its differentiating factor to see whether the bacteria can ferment lactose or are non-lactose. Measurement of cell mass. You want to determine the cell mass by direct counting. You can tell me that. You can tell me that there are how many cells in the sample, right? By direct counting. You can do that, right? If you take a sample, you do direct counting, you can tell me how many cells you have in your sample, right? But that doesn't tell you the mass. Mass means the weight, or in terms of carbon, how much is it in weight? There are several ways of doing it. The hard way or the easy way? Easy way is done. Every cell you times 20 femtogram carbon. Easy. But it's not accurate. Not very accurate. But roughly it's what it is. So this figure is from researchers who spend many hours counting the cells and measuring the size, and then coming up with this factor, conversion factor. Okay, so from here you can get the cells in gram. So that's your mass. The harder way is to measure the biovolume yourself, which means you measure up to 300 cells. 300 bacterial cells. You have to measure the diameter, the length, and you calculate the biovolume, the volume of the cell, one by one. And then you estimate the mass. So this is the proper way. This is the shortcut way. Another way is by not doing this at all, by using proxy. Do you know what's proxy? When you cannot attend a meeting, you ask someone to attend on your behalf. So that person becomes your proxy. So we want to measure biomass, but we can't measure it directly. So we measure something else that can be related to the biomass as a proxy. Okay? So for bacteria, usually we measure phospholipids. Okay? For phototrophs, usually we measure chlorophyll A. For fungi, you can measure either chitin or ergosterol. So these are suitable proxies that tell you the biomass of or the microbes that you're studying. Okay? So all this can be proxies for biomass. Okay? For bacteria, it's phospholipid. For phototrophs, chlorophyll A. For fungi, either chitin or ergosterol. Ergosterol is better.